Okay, so the last little part of the cycle we're going to talk about is the urea cycle and then the fate of all of these amino acids. So the urea cycle is what actually removes ammonium from our bodies. So ammonium is actually toxic to our bodies. So it stays in the liver, and in the liver, it undergoes the urea cycle to convert ammonium to urea. I keep saying urea wrong, weird today. Okay, but overall, what this process does is it takes two ammoniums and a carbon dioxide and it converts it into urea. So that's this molecule here. This is a molecule that is in our urine, and that's how we remove it from our bodies. So we make urea plus more hydrogen, H plus, and some water. So we produce about 25 to 30 grams of this a day on average. Okay, so let's look at the citric acid cycle specifically for all of our different amino acids. So each carbon skeleton of each amino acid is a little bit different and overall has a different place that it enters the citric acid cycle. So depending on its structure, it could enter as pyruvate, or it could enter way later in the system, like phenylalanine and tyrosine enter as fumarate once they get to that position. So it's not as straightforward to add up the ATP. So we're not going to worry about how much ATP is produced in each of these, because then we'd have to go back and look at every single step after it gets added in. And it gets real tedious real fast. So use this kind of chart to see in terms of kind of what it forms is more of the questions that we're going to be asking. So let's do a learning check on that. We're going to have to go back and forth for this, but match each intermediate. So we're going to focus on the citric acid cycle. So match the intermediate in the citric acid cycle with the amino acid that provides its carbon skeleton. So this is how these questions are going to be asked about this particular thing. So pyruvate, and fumarate, or alpha ketoglutarate are our options. So cysteine, which intermediate does it enter? So let's look back at our chart up here, and we see that cysteine is right here. So it enters as pyruvate. Okay, then we have glutamine. We'll have to go back up and look and find, glutamine is here, and that enters as alpha ketoglutarate. Tyrosine is over here and enters as fumarate. And our last one is serine. So if we go back up, we see serine again is over here, enters as pyruvate. So that finishes up how our proteins are degraded. Long story short, it's very complex, and because all of the different structures are so complex.